Hello everyone, this module is brought to you by the MTMV instructors of Angeles University Foundation. My name is Romania Salazar, your module writer for Module 6, which is all about DNA viruses. At the end of this module, the student should be able to first identify and differentiate the DNA virus classification according to their structural characteristics. And second, is for the students to be able to differentiate these DNA viruses according to the disease they cause and its clinical manifestation. To accomplish these objectives, our topic includes an introduction that will include all the characteristics of DNA viruses that is unique to them alone, and one by one we will discuss herpes viridae, hepadnaviridae, parvoviridae, papaviridae, adenoviridae, and poxviridae. We have mentioned from our previous lesson that a virus should have the basic parts. A virus should always have a protein component called capsid and a nucleic acid or the viral genome that could either be a DNA or RNA. Now, classifying the different viruses are based on this following: nucleic acid present, viral symmetry, and presence or absence of an envelope. DNA viruses are viruses whose nucleic acid component is a DNA. And based on symmetry, we can classify them into two: icosahedral DNA viruses and complex DNA viruses. Furthermore, your icosahedral DNA virus can be classified into being envelope and non-envelope DNA viruses. For the envelope, we only have double-stranded envelope icosahedral DNA viruses, and they are herpes viridae and hepadnaviridae. For the non-envelope icosahedral DNA viruses, we have single-stranded and double-stranded. For the single-stranded, we have parvoviridae, and for the double-stranded, we have papaviridae and adenoviridae. Now, for the complex DNA viruses, we only have envelope, double-stranded, and it is poxviridae. The first group of DNA virus that we will discuss is the herpes viridae. Note that the virus under the herpes viridae family are icosahedral in symmetry, they are enveloped, and containing double-stranded DNA viruses. The herpes viridae family contains several important human agents. They are herpes simplex virus type 1 or the HSV1, herpes simplex virus type 2 or the HSV2, varicella zoster virus or the HHV3, Epstein-Barr virus or the HHV4, cytomegalovirus or the HHV5, human herpes virus type 6 or the HHV6, human herpes virus 7 or the HHV7, and finally, human herpes virus 8 or the HHV8. One of the most notable characteristics of this group is its ability to cause lifelong potency or persistence in the host, which can undergo periodic reactivations. This is called viral latency. Viral latency is the ability of the virus to remain dormant within the host cell, which sometimes can establish a lifelong infection. Depending on the virus, the trigger of latency is highly variable. The first group under the herpes viridae family that we will discuss is the herpes simplex virus. There are two distinct herpes simplex viruses. One is your HSV1 or the herpes simplex virus type 1 and HSV2, which is your herpes simplex virus type 2. They are almost similar in characteristic, but classically, type 1 is spread by contact involving saliva, while your type 2 is commonly transmitted sexually or maternal, meaning a transfer from the mother to the fetus. These here are the following diseases caused by herpes simplex virus. First is your oral herpes. This is caused by herpes simplex virus type 1, which is commonly seen in intraoral mucosal vesicles or ulcerations in the buccal mucosa, or sometimes in the posterior pharynx and gingival mucosa. Next is your genital herpes. Your genital herpes, on the other hand, is usually caused by herpes simplex virus type 2, which appears in females as vesicles in the mucosa of the labia, vagina, or both, and in males naman in the shaft, glans, and prepuce of the penis. Your neonatal herpes usually are infection acquired in utero, intrapartum, or postnatally, but it may be caused by both HSV1 and HSV2. But if it is caused by your herpes simplex virus type 2, it is more likely to be severe. Next is your HSV encephalitis. This is a rare but devastating disease, usually caused by your HSV2 in neonates and caused by HSV1 in children and adults. And lastly, you have your ocular herpes. This is an infection in the conjunctiva which is manifested by the swelling of the eyelids with vesicles or ulcers. Next is your varicella zoster virus. This is spread by the droplet or by inhalation or by direct contact with the infectious lesion. It can be manifested into two different clinical manifestations. One is varicella or the chickenpox, which is the primary infection, and the other one is zoster or shingles. This is due to reactivation of your varicella zoster virus. Your primary infection is characterized by generalized vesicular eruption of the skin and mucous membrane, and usually the fluid-filled blisters can be seen in different parts of the body, and most likely in every parts of the body. While in the reactivation of your varicella zoster virus or shingles, the vesicular blisters are limited in the skin area, innervated by a single sensory ganglion. So basically, in one portion only you can see them, most commonly in the trunk or arms. And since it's sensory ganglion which is affected or involved, it should be very painful. The next virus under herpes viridae is the Epstein-Barr virus. This is the causative agent of acute infectious mononucleosis. The virus is also associated with several diseases such as nasopharyngeal carcinoma, Burkitt's lymphoma, and Hodgkin's and non-Hodgkin's lymphoma. They are transmitted by infected saliva, which is why it is commonly known as the kissing disease. And it is also initiating oropharynx infection. Another virus under herpes viridae is your CMV or cytomegalovirus. This is the common cause of congenital infection that can result to several abnormalities because the virus specifically replicates in human fibroblasts. They may affect a variety of hosts as well. They can affect normal adults, immunocompromised hosts, and neonatal hosts or babies. And because of this variety of hosts, the manifestation of the disease may be different from each of the hosts. So in normal hosts, it may appear asymptomatic or occasionally may cause an infectious mononucleosis-like infection and sometimes subclinical hepatitis. In an immunocompromised host, on the other hand, may exhibit pneumonia, colitis, retinitis, or hepatitis, or cause disseminated infection and generalized leukopenia, which means decreased WBC count. In neonatal or infants, they may cause fetal death. They may also cause cytomegalic inclusion disease, which is a generalized infection of the infants, exhibiting jaundice, hepatomegaly, thrombocytopenia, microcephaly, pneumonitis, several CNS damage, and retinitis. Next is your human herpes virus type 6 or the HHV6. They have two variants, type A and type B. Although they cannot be differentiated serologically, they are a common pathogen. That is why 94% of young adults are seropositive with this one. Modes of transmission includes inhalation of the respiratory droplets. Now, this is associated with childhood diseases such as Rosola. Other name for Rosola is Rosola infantum, exanthem, and the sixth disease. Next is your human herpes virus type 7 or the HHV7. Also, it causes Rosola that is clinically identical with that of the herpes virus type 6. Although CD4 is the molecule that serves as a receptor for this virus. That's why, because it infects the T lymphocyte, it is like your HIV virus. Next is your human herpes type 8 or the HHV8, which is also known as the Kaposi sarcoma-associated herpes virus. It can be detected in all forms of Kaposi sarcoma, which is a common infection associated among immunocompromised individuals and patients like those with AIDS and HIV. Now, another family under icosahedral envelope double-stranded DNA viruses are the Hepadnaviridae. So Hepadnaviridae is a causative agent of your viral hepatitis. But take note that not all hepatitis viruses are DNA viruses. Actually, it is only the hepatitis B virus that contains DNA as its viral nucleic genome. Now, hepatitis B virus is a blood-borne pathogen, meaning it is transmitted via blood, which is the causative agent of your serum hepatitis. Serum hepatitis is a systemic disease, which is primarily affecting your liver. However, lower concentration of the virus may appear in semen, vaginal fluid, saliva, and other fluids such as urine, tears, sweat, breast milk can also contain hepatitis B surface antigen, but they do not seem to be infective. Okay? They have this hepatitis virus in other fluid, but they are not infective. Main modes of transmission are sexual transmission, perinatal, which is from your mother to the baby, and parenteral, which could be from sharing of needles, and blood transfusion. In the course of the study of hepatitis infection, it is very important to understand the structure of the hepatitis virus because it significantly influences the interpretation of serological tests upon the diagnosis of the different stages of the disease. If you look at this illustration in the slide, you will see that the hepatitis virus At the center of it is a DNA polymerase, which is made up of double-stranded DNA. And on the surface of the virus are antigens. These antigens induce immune response once the virus enters the body. So, with this virus, we have three important antigen: the HBC antigen, the HBs antigen, and the HBe antigen. HBC antigen, or the core antigen, since it is found in the core of the virus, are not detected in the serum. HBs antigen, found in the surface, or the surface antigen, it serves as the marker of infectivity, meaning when you found this in your serum, or the individual is positive with HBs antigen in the serum, it would mean that the antigen is in the body. Therefore, the virus is in the body or present in the body. Third is your HBe antigen. This is found in between the core and the surface. It is found to appear early in the acute infection. Kaya nga siya E, which means early. It marks the repetitive stage of the virus, meaning it serves to signal that the body is harboring the virus. Therefore, it is infective. Also, at the same time, when your HBe antigen is present, it is an indication that the virus is replicating. Therefore, the infectivity is high. Now, once the virus have entered the body, as a response, syempre, your body will produce antibody to counteract this antigen. That is why it is expected upon the exposure of the body from this antigen that your body will produce corresponding antibody against this antigen. And these antibodies are the following. So, you have your IgM, anti-HBC, IgG, anti-HBC again, anti-HBs, and anti-HBe. So now, your anti-HBC IgM appears early on acute infection. Remember that the IgM is a type of immunoglobulin that appears in early or acute infection. On the other hand, your IgG appears in chronic or past infection. Next is your anti-HBs. This is an indication that your body has elicited immune response, whether it might be from an infection or merely from vaccination. Take note that during these two, vaccination and infection, the actual HBs antigen enters your body. Thus, your body produces antibody against it, and as expected, your immune response will produce an anti-HBs. Therefore, because of this, your anti-HBs can denote two things. Either the person is currently infected with the virus reacting to the surface antigen, or the individual has reacted to the HBs antigen introduced through vaccination. Okay? And the fourth one is the anti-HBe. So the last one marks the end of replicative stage of the disease. Remember that your HBe antigen confer high infectivity. Thus, producing an anti-HBe means the end stage of the disease at its mark. Let us look at this chart to further help us master the interpretation of hepatitis serological markers. So this side of the chart marks the possible assay results combination, and this side states their respective interpretation. So let's take a look at this first. If your results give a positive HBs antigen and a negative anti-HBs and negative anti-HBC, remember that from the previous slide, your HBs antigen is a marker of acute infection, and since no antibodies were produced yet because they are still negative here, it is an indicator of early acute infection. Next is when you have a positive HBs antigen, positive anti-HBs, and positive anti-HBC. This is an indication of hepatitis B infection, either acute or chronic, that is differentiated with either IgM or IgG. Remember again from the previous slide that IgM is indicative of acute or early stages of the infection, while your IgG is an indicative of chronic infection. Now, when you have a negative HBs antigen with positive anti-HBs and positive anti-HBC, this could be an indicative of previous hepatitis B virus infection and immunity to hepatitis B. However, if you have a negative HBs antigen and a negative anti-HBs rather, with positive anti-HBC, this could mean few other situations. Either there is a low level of hepatitis B virus whom it could be from a carrier, or a window phase between the disappearance of the HBs antigen and the appearance of anti-HBs. It might be recommended to further analyze which among the anti-HBC is present to help validate the infectivity. Furthermore, if you have a negative parameters for HBs antigen and anti-HBs and anti-HBC, this could mean the individual has never been infected or has any history because there is no marker for hepatitis in the serum. And lastly, if you have a negative HBs antigen and negative anti-HBC with a positive anti-HBs, this would mean a successful vaccination. Why? Take note that the vaccine given is made up of an HBs antigen. Therefore, if you do not have HBs antigen in your serum, but still you have the anti-HBs, it could mean that it is an immune response of your body towards the vaccination that was given to you. Next on our discussion is Parvoviridae. These viruses are icosahedral, non-enveloped, single-stranded DNA viruses, which are the smallest DNA animal virus. One of the most distinct and notable pathogenic virus in this family is the Parvo B19. Parvo B19 has a tropism for erythroid progenitor cells, meaning it has the affinity or attraction to cells that give birth to erythrocytes or the red blood cells. The virus is the one causing fifth disease or the erythema infectiosum, which appears to be common in children and known to produce a slap cheek presentation as seen in this picture. The virus can be found in blood and respiratory secretions of people infected with it. And this can be transmitted via respiratory route or parenterally by blood transfusion or of infected blood products, and vertically from mother to fetus. Next is the Papoviridae family. This family are icosahedral, non-enveloped, and take note this time double-stranded DNA virus. And just a nice to know trivia for this family. The actual name of this family are coming from the members of the virus under this family itself. The Pa from papillomavirus, Po for polyomavirus, and Va for vacuolating agent which is the effect of SV40, a primate virus that causes vacuolization of infected epithelial cells. However, for the purpose of discussion today, we will be only discussing papillomavirus and polyomavirus, which is the one infecting humans. Your papillomavirus causes papillomas or warts. They are also associated with HPV or human papillomavirus that is associated with cervical cancer. Polyomaviruses, on the other hand, mainly affects immunosuppressed individuals. It can cause progressive multifocal leukoencephalopathy or PML and ureteral stenosis and hemorrhagic cystitis. It has two subgroups or etiologic agent. It is the JCV or the JC virus that causes PML and BK or BKV virus that causes urinary tract diseases. In this picture, JCV is involved. In PML or progressive multifocal leukoencephalopathy, wherein the brain gradually loses its myelin sheaths or white matter. So as you can see, the normal brain doesn't have this dots, green dots, which represents infection via the JC virus. This picture below represents nephritis or inflammation of the kidney, particularly the glomerulus caused by the BKV. Another icosahedral, non-enveloped, double-stranded DNA virus is Adenoviridae. These are naked viruses that can replicate and reproduce in the respiratory, gastrointestinal, urinary tract, and even in the eye. But however, most of the diseases or infection are subclinical and require no specific treatment. They may be transmitted through aerosol, fomites, oral fecal route, or even just personal contact. All human adenoviruses are classified in the Mastadenovirus genus. They have 49 distinct serotypes and 6 subgenera. Subgenera B, serotype 3, 7, and 14 causes pharyngoconjunctival fever. Under the same subgenera, serotype 7, 14, and 21 causes acute respiratory diseases. Subgenera B, serotype 3 and 7 causes pneumonia. Subgenera C, serotype 1, 2, 5, and 6 causes acute febrile pharyngitis, specifically in children. Subgenera D, serotype 8, 19, and 37 causes epidemic keratoconjunctivitis. Your subgenera E, serotype 4, causes acute respiratory disease with pneumonia and fever. And lastly, your subgenera F, serotype 40 and 41 are known to be the enteric viruses, which causes gastroenteritis. The last family on the DNA viruses are the Poxviridae. Poxviridae are complex, enveloped, double-stranded DNA viruses. It is the largest and most complex of the DNA viruses infecting humans. And most of the infections caused by pox viruses are characterized by rash. Under the electron microscope, it appears to be particles having a shape of a brick or ellipsoidal particles. The different infections caused by Poxviridae include smallpox, monkeypox, cowpox, and orthopox. Most of these have similar presentation, which is the formation of rash all over the body or specifically on affected areas only. And on the following slide, we'll be taking up each of these infections. Smallpox is considered to be the world's most devastating disease known ever to humanity. It is caused by the variola virus, which is the first virus to be eradicated from Earth. It was in 1980 when it was eradicated, following a global immunization initiative. Although up to now, it is a potential bioterrorism agent because the human population today has little or no immunity against variola virus. The smallpox is transmitted via infected droplets during close contact. But we have vaccines for smallpox. But the vaccine contains vaccinia virus instead. Vaccinia virus is a virus that is used for smallpox vaccination and serves as a laboratory model for pox viruses. It is considered as the prototype of variola virus. Well, in short, this is what the vaccine is composed of. Monkeypox is a rare animal disease caused by monkeypox virus. This was first recognized in captive monkeys in 1958 and then eventually recorded the first human infection in early 1970s, specifically in West and Central Africa after the eradication of smallpox from those regions. It is acquired by direct contact with specifically wild animals that are killed for food. Vaccinations with vaccinia either protect the individual against monkeypox or if they get infected, the severity of disease is less. Now, cowpox is a disease of the cattle which is milder than the pox disease of other animals. Similarly, this infection in human occurs by direct contact during milking, and the lesions in the milkers or milkmaids are usually confined on the hands. Edward Jenner observed that those who have had cowpox are immune to smallpox. So Edward Jenner is the one who initiated the vaccination against smallpox. And lastly, orthpox is a disease in sheep and goats that is prevalent worldwide. These are transmitted to humans by direct contact with an infected animal. It is considered to be an occupational disease of people handling sheep and goats. These are my references. For your questions and inquiries, please feel free to contact me, email, or chat me with any of this platform. Thank you very much and happy learning.